Okay, so I don't know if you guys noticed that I had posted some changes to my schedule. I may or may not told you guys that I changed that. So make sure you take a look at that. That's different from the one that was at the beginning of the semester. You'll notice that all my office hours are scrunched on Monday, Tuesday, Wednesday. There's a stupid reason behind that, so I'm not going to explain it because it really is stupid. But know that pretty much I'm here every day from 8 to 3. And if I don't have office hours and I'm not teaching class, you can still contact me and I'll still work with you within those hours. It's best, obviously, if it's not during office hours, to contact me ahead of time if you need to meet with me. Does that make sense to you guys? Okay. And so I think we almost finished chapter 1, right? And we don't have a whole lot on our site. I did get my book. Yay. I'm so excited. And at this point, most of you guys have had to pay, right, for the Connect, right? Your trial would have expired. So please make sure and do that so you can do the homework that I'll be posting. We've already done chapter 1, but there'll be a couple of chapter 2 homeworks. I put it on reserve at the library. The new library is so nice, y'all, if you haven't been in there yet. Building 7 right behind. Yeah, it's so nice. I honestly was never in the other one. I got hired right after Katrina, the year after Katrina, so I never went in the old one that used to be literally in the same spot the new one is. I just saw the decrepit, poor building that Katrina left behind. But it's really nice. It's really nice. So definitely go in there. And then at the study lab, I also put a book there. So if you're in the study lab studying, like maybe with Erin when she's in there, you know, yes. Yeah. For in the study lab, if you need to use a book, you got to give them your ID, and they'll give you your book so you don't steal the book. So you got to ask the tutors for the books. But they're in there. So I updated those. So I just want to, I think we were just about, just at the very end of the chapter 1 lecture, but forgive me if I'm wrong, right? Pretty much the 3 different main fields. So transmission genetics, molecular genetics, and population genetics are the 3 main fields that genetics are divided up into. Angela, I know you're here, but please sign the pad so I don't forget to mark you. So the other thing I was going to say, now that I have my book and I can easily flip through it instead of having to wait for pages to load on my computer, I will finish the syllabus and hopefully have it for you guys on Thursday. So you guys will know where we're going next. But, you know, clearly we're on chapter 2 next. I had told you guys that and posted that. So the book kind of is divided up in that way too. They start out with transmission genetics. That's what we're going to start out with. Then they go on to molecular and population. So you need to be able to know in general what each one of these is. So transmission is the oldest field of genetics. And that's where they just examined how traits are passed from one generation to the next. So this is good old Mendel and his pea plants, and we'll start doing Punnett squares again today. And so, you know, this is before he knew the molecular aspects that we know today, right? He knew that there were these discrete units. We refer to those as what now? What's that term we use for a segment of DNA that codes for something? A gene. Hence, genetics, right? It's in the name. We're not talking about blue genes, right? We're talking about a gene. A discrete segment of the DNA that codes for something. So they did basic experimental crosses where they crossed different individuals, and they analyzed it over several generations. And they could do this quantitatively. They literally count. He counted how many came out purple flowers as opposed to how many came out white flowers, and figured out, you know, this pattern of inheritance based on what he observed. So, of course, Chapter 2 is Mendelian patterns. Chapter 3 is chromosomes and their roles in inheritance. And, yep, sign the book. And then chapters 4 through 8 go over different things like sex-linked and non-Mendelian patterns of inheritance. So we probably won't completely cover 3, and we'll jump around to some of those, some of the topics in 4 and 8. We won't cover whole chapters after Chapter 2, really, as far as transmission genetics goes. And so then we have molecular, which is, of course, the most modern, right, since we've had the ability to be able to literally sequence DNA. So I found in my office the Human Genome Project poster that I used to have on my windows to my office. But what I've done is I've put a little sign on it, so hopefully nobody takes it or throws it away. But you'll notice we have a bulletin board in this classroom. So I'm going to come and hang it up in here so we can look at it before and after class or even during class and actually see that. So we already know, right, that genes are organized and we know how they function. We can analyze right down to the code level for DNA and RNA and proteins. We can figure out what amino acids make up the proteins. Proteins, on the other hand, are not quite as simple as nucleic acid, right? Protein science is like a huge area of biotechnology. Because although we can synthesize proteins, like how and why they fold the way they do is still very much a mystery. So there's lots and lots of research done in proteins nowadays, more so than DNA. Like, if you could crack that code, like, if you could come up with a computer program, you'd be a gazillionaire. Because we just don't know. We don't know the code for proteins. You know, it's very complicated. So, molecular genetics typically employ a genetic approach to research, and they study mutations, where they actually purposely cause changes in the DNA to see what effect it has on the organism. So does that cause them to lose function? And we know a lot of the diseases that we talk about are a loss of function, where the mutation in the DNA no longer produces a functional protein, right? And so that person can't do a particular thing. So what did PKU stand for that we talked about last time? What is that defect? It's the one where you can't have aspartame because you can't convert what amino acid, though? What's the P? Phenylalanine. Yeah, phenylalanine. You can't convert it to, I think, tyrosine. Yeah. And so you need to limit it in your diet. Basically, don't eat it if you can avoid it because you'll build up too much phenylalanine, and that can cause damage to the brain. Right? It'll affect the brain. So there's a lot. Most of the diseases that you look at that are genetically linked, that's what's wrong, is that the DNA is got a mutation in it, and it doesn't make a functional protein anymore. And so those people lose that ability to do that particular function. And depending on how important that function is, you can maybe change with diet, right? You may supplement with drugs like insulin, or other therapeutic drugs to kind of put a Band-Aid on the problem, right? Especially with cystic fibrosis, they really just put a Band-Aid on the problem. And that's one of the things that we'll talk about too. So lots of chapters, right? And again, a few of them will cover whole chapters. Others we're going to kind of skip around. So I'll have that full list for us pretty soon. And then the last is population genetics, which unfortunately we usually run out of time for. So I think maybe this semester we'll maybe do some independent projects on population genetics. So I have an event that I usually do every spring that we're going to do every semester now. And it used to be called the Science Expo. but now it's going to be called STEAM Day at Delgado, standing for Science, Technology, Engineering, Art, and Mathematics. So there's going to be a lot of divisions, and basically it's a science fair, and local area 5th and 6th grade students are going to be invited to come. And so you'll have a table, and you can put together a poster board or some type of, I prefer interactive activities to do with the kids that come by. So what I would like for us to do is try to get some groups together. It'll be bonus, right? And put together something for our genetics table, if you guys are interested in doing that. I'll put together a discussion board and stuff, and we'll start building ideas and figuring out what we might want to do. But I think the topic we'll pick is population genetics, because again, that'll give us a chance to research this. And I think it'd be something that would be of interest to the kids at that level, right? So what do we mean by that? So population genetics deals with genetic variation within populations. So I mean, we could very easily do something like talk about blood typing, right? And how that's genetically inherited in its distribution among the different populations and ethnicities in our world, right? We don't even have to limit it to our country, right, on this planet. And there's actually some really cool stuff like in Japan, and there are some people too, like they'll eat particular diets based on their blood type. I don't know how much validity is to that, but in Japan too, they actually will decide who they will mate with or partner with based on blood types. And actually, they say that your blood type, you know, affects your personality. I don't know how much of that is true either. But I don't know. I mean, there's obviously probably some, you know, basis somewhere to being left-handed being genetic. You know what I'm saying? You know, because you're programmed a particular way. So yeah, I mean, it could be genetic. Whether it's like a trait that's like, I would say it's one of those things that's multiple factors that come together for that one particular trait. Does that make sense? Right? So it's not just one thing. It's, you know, how your muscles formed on your right, your left, or the bone structure. I mean, all those things are controlled by so many genes. You know what I'm saying? So I think it's just coincidence sometimes too, where, you know, you'll have several left-handed people in the same family too, you know. But, and then, you know, when I was a kid, if you were left-handed, they made you be like right-handed, and then you became ambidextrous, right? My son's left-handed, but both me and my husband were right-handed. My current husband's left-handed, which is nice because I have no idea about the left-handed world, and so they help each other out. And they teach me. And I try to be better for my students, you know, and understanding that, you know, there are lefties, and that's all right. Except most of this world is made right-handed, which makes it difficult sometimes. So population geneticists develop mathematical theories to explain the prevalence of certain alleles within populations too, right? Why do we have so many A blood types versus B and AB and things like that? So population genetics, of course, is at the end of the book, 3 chapters. So, and again, you know, this starts talking into too, well, you know, even though you'll have identical twins, over time, the effect of environment and their own choices, they can look different, right? If one of them eats nothing but junk food and the other one sticks to healthy food, they're going to look different. Even though genetically they're the same. It's the effect of that environmental choice that will change their bodies. And of course, this factors into evolution, you know, over time within a particular population. So that's something too we could look at. You know, we can do a poster on the evolution of a particular species, like the change that we talked about in horses. I love horses too, so I thought I was going to be an equine vet when I was in the 8th grade. I changed my mind clearly. All right, so then that brings us to Chapter 2, which of course is transmission genetics. And most of what we know about genetics started with Mendel. But before that, there are some interesting theories out there on how it was that we came to be and look the way we do. So there is the theory of panogenesis, the theory of preforminationism, the blending theory of inheritance. So panogenesis was proposed by Hippocrates before Christ. He said that seeds are produced by all parts of the body, right? So imagine this, your brain produces a seed, your heart produces a seed. They're collected in reproductive organs, so all these seeds, right? Your heart seeds, your brain seeds, your eye seeds are all collected in your reproductive organs. And then transmitted to the offspring at the moment of conception. You know, we try to understand what we can't see, right? My English professor, when I was in high school, like sophomore year or something like that, we had to write papers all the time. Mine was about how God was everybody's answer to their unanswered questions. It is. Right? So he was like all concerned, you know. And I was like, yeah, I had a moment of atheism. I think we all have that, you know. It happens. But, you know, it's okay to believe or not believe. Right? But he was really concerned for me. So I had. But in truth, really, right, for most of us, we try to explain what we don't understand. So the theory of performationism, this one cracked. I love this one. This one's so funny. People using early microscopes thought they saw a miniature human or homunculus in the sperm. They thought they saw a little person in the sperm. So this is how much of a nerd I am. When I was in graduate school, we had a bowling team. Our team was called Homunculus. No one else understood.
So when I think of that word homunculus, I think alchemy.
Yeah. It does sound like an alchemy term. So, with proper nourishment, the homunculus unfolds into its adult portions. So again, I mean, they believe that, like, you basically came from your dad, and you just, like, grew in your mom.
Just, like, super tiny and then...
Yeah, it just unfolded.
But then you, like, became a baby? Like, you were, like, a little man, and then you, like, grew into a baby?
I don't know. So spermists believed that the characteristics of the future person were found in the sperm only, right? So basically just in the sperm. The ovalists believed that the characteristics of the future person resided actually in the egg, right? So there was some debate, you know, which one had the little person.
That's so funny that it was like a real theory.
Yep. To us, you know, we laugh, right? We're like, right? To them, I mean, again, they're just trying to make sense of it. And then there was the blending theory, which probably came closest to somewhat what we deal with today. So they believed that the factors that controlled hereditary traits were malleable, like you could mold them. And they could blend together to form generation to generation. So I'm trying to think of an example of that. You know, it kind of really makes sense. Oh, I forgot that picture for you guys. Especially when you see siblings, like I said, where you end up with ones that are vastly different from the parents, right? You could clearly see it's more of a mixture, and it doesn't seem like that these things are discrete and separate. I don't know a good example for that one. But it wasn't until Mendel, working with his pea plants in the monastery in his little plot, right? This man was meticulous and amazing observer of science. He refuted all of those previous theories. So for us, for this chapter, the objectives that we want to try and reach is that we want to be able to explain the characteristics of the pea plants that made them a suitable organism to study genetically. So why did he basically, y'all, he got lucky, right? These are relatively simplistic plants, and there wasn't a whole lot of variety, right? So he really got lucky. And so we'll talk about the aspects that made it really suitable. Outline the steps that he followed to make crosses between different strains of pea plants, and be able to list the 7 characteristics of pea plants that Mendel chose to study. And again, he got lucky with these. They ended up being pretty discrete units as far as the DNA went. So he's considered, of course, the father of genetics. Again, this is not a history class, but this was back in the early 1800s. Pretty amazing stuff. He had experience working with plants, grafting trees when he was younger. So probably, when he became a monk and started investigating plants further in this inheritance, it was familiar to him, right? He had a familiarity, he had an interest. And grafting trees is not easy. And so you had to really be precise, and you really had to, have an attention to detail, which definitely really helped him in his studies. At university, he studied physics and natural history. Again, these disciplines really helped prepare him for doing the excellent job that he did. Especially in physics, we have all these laws and orders, and, he thought of things, it's got to follow a certain progression, right? It isn't just random. And there's got to be some order to it. I'm going to find it, right? I'm going to find an explanation on why this happens. And so, of course, these laws can be stated mathematically. So he was an Austrian monk, and he only, I wanted to actually bring my tape measure and map this out, but I think these are 1-foot tiles on our floor. So imagine, right, 100, if we went down the hallway and counted 150 of these by 23, or even this classroom. It isn't much bigger than this classroom, probably, what he worked with. He didn't have fields upon fields like they do out in the sugarcane fields that we drive by when we get out of the city, right? He had this little plot of land, and boy did he put it to some serious use. He performed thousands of crosses over several generations. And this is what made it all possible. He took meticulous notes. Any scientist nowadays would be envious of the level of detail of his notebooks, and in his quantitative analysis and his mathematical analysis of what he saw. So he published his work, Experiments on Plant Hybrids, in 1866, and it was largely overlooked by most of the people in the scientific community. Title alone, right? How many of us, you go into the library and I know my son picks it by not even the title, it's the picture of the book and how many pictures are on the inside right now, right? But, you'll pick up a book and, just the cover page, this isn't interesting, move on to something else. So for a lot of people, they were like, it's not of interest to me, right? So vastly overlooked for some 34 years. And again, the title really didn't draw us to it, right? How important and amazing it was. And this was all before the lack of understanding of chromosomes and transmission. So later, his work was rediscovered by 3 botanists working independently. So they, of course, botany, so of course his title, plants, hybrids, they're going to look at it, right? Unlike other scientists. So Hugo in Holland, Carl in Germany, and Erich von, God help me with that last name. I think the T is actually silent, of Austria. It's a ch sound? Yeah. So hybridization, what does that mean? Right, so we're going to use a lot of terms. You might even want to keep a section of your notebook just for terms, right? Just to help center yourself and get you through it. So hybridization is the mating or crossing between 2 individuals that have different characteristics. Yeah, I'm good. Right?
That applies for animals too.
It applies to humans even. Right? So when white people, Caucasian, right, and African American, when they mate, right, that you could consider it a hybridization because they're vastly different traits, definitely in skin color. And then, of course, you get a variation, too, in the children that's produced. You'll get some children that are really dark skinned, right? Others that are really light and even lighter, right, and shades in between. And it just depends on what genes were passed from which parent, right, and how they're expressed. And there are some traits that are very strong and more dominant over others. Like, for instance, Asian traits for their facial structure are very dominant. So usually if an Asian person mates with another person, the children look very much what the features what we consider Asian, right? The eyes, the way they're shaped in a particular way.
So I have a question. Okay, for example, see, two parents all both mixed of mixed race. What does the child be considered? See, you have mother is half mixed black and Italian. And
Yeah, some people come up for names for mixes of ethnicities. I don't particularly tend to use them, but I've heard mulatto is a mix between African American and Caucasian. I think some people take offense to that, but the truth of the matter is, there are these distinct differences because at one time those communities were all in one place. And now we have so much mixing. I mean, if you look at my background, granted it's mostly Eastern European, but German, French, American Indian. You name it and I probably got it. Scandinavian, German, French, Scot-Irish. I just call myself a mutt or a true American. I'm not a purebred by any means or stretch of the imagination. Right? So it's like I get kind of annoyed too, like those ethnicity things, like Caucasian, really? Why do we write these things? Well, because we want to track, right, these differences within the population. That's all that is. We're not trying to offend anybody, right, upset anybody. We're just trying to track information. Does that make sense? Right, so it makes it really simple when you talk about dogs, right? The different breeds of dogs. But they're all the same species. But they have these really big varieties. We were literally driving down the road the other day, and someone had their dog hanging out the window like you're not supposed to do, right? Something could whack their head off. But there was this really cute dog, and I was like, oh, it's a miniature pinscher, or, and he's like, oh, no. So we pull up next to the guy, and of course my husband has to roll down the window and ask the guy, what kind of dog is that? It was a miniature pinscher and a Chihuahua crossed. I got one of them. But, how did you even end up with the miniature pinschers, right? Or they have miniature, like, Dobermans now, apparently. That's just wrong. Yeah. Yeah. Oh, I mean, they've mixed everything up. And then, like, now it's, like, really popular to have a mixed dog. Like, for me, I always get rescues. They're always mixes. They're the best dogs. My husband's got a pug. Yeah, you need a hair dog. Yeah. Yorkies are... I loved my Yorkie when I was a kid. They're awesome dogs. But, yeah, no... but like, it's actually good. I'm glad that it's becoming popular to mix breeds, because inbreeding, like, generation after generation, you're keeping those same traits that you like the particular color or the size or the shape or, all those fun things, right? But then you're also sometimes amplifying the bad genes, right?
I was watching that documentary, and they were saying that, like, you have to be careful because, like, terriers are really aggressive, and then, like, if you mix it with another aggressive breed, you get, like, double aggression.
Right, yeah.
And the dog is just like...
Yeah. And some of those things, aggression and stuff like that are genetically linked, too. But it's also how you raise a dog, and how you treat it. And some of them are just psycho. There's nothing that you can do, I think. I don't know. That's also why I try to get my rescues very young. So, like, if they have any bad habits, I can hopefully break those. But even my husband's dog, who's a purebred, spoiled little brat, I taught her to go to the bathroom outside most of the time. This morning being the exception. At 4 a.m. when she's got to go, she's going to shit on my floor. I don't have a doggy door. But, like that breed, they have really bad allergies. I mean, she has the worst allergies. She has to take medicine every day. There are ones that were hip dysplasia, because of inbreeding is really bad. So, again, like, I'm glad that it's popular now to mix breed. It's a mixing up of the genes, right? But again, you want to try and, like, match good ones. I mean, my first dog that I lost about a year or a half ago now, he was a Border Collie Chow, Teddy Bear Chow Border Collie mix that my mother-in-law had. She took in a stray lab puppy, she thought, from the swamp, which, again, he couldn't even been full lab. He might have been a mix. We don't know. Got her dog pregnant because he wasn't a puppy anymore. And then I ended up with Blue, who I picked out when he was 10 days old. But I consider him a rescue because he was a rescue of a rescue, you see. Right? And he was awesome. I mean, he had all the good traits of all those breeds. He was just amazing. My current dog, she's some type of lab mix. Again, a rescue I got from a rescue organization. She's awesome. I love her. They're like, what is she? I'm like, perfect. And she looked a lot like my old dogs. When they brought her out, my husband was like, oh, no. She had the curled tail, and I love that. They don't hit you with it. Love the curled tails. I had a roommate that had a greyhound. That is a whip. That should be a registered weapon on the back of a dog. That's painful when you get hit with that thing, right? So, yes, she's awesome. Us mixes are awesome, right? Because, you can hopefully dilute out the bad traits and amplify the good ones. So he very simply, saw that in the pea plants you had either a purple flower or a white flower. So what happens if I breed these? What's going to happen, right? So he generated hybrids. These are the offspring, right? So my dog is some type of hybrid, right? She's a whole bunch of breeds. If I was rich, I'd send it off for genetic testing. Nowadays, you can do that, right? That what is it, Me 23? 23 Me? Yeah, it used to be $100. It's like $200 now. So
It's cool though.
Yeah.
I did it for my boyfriend for his birthday.
yeah,
It's really cool.
Kendra Onspa did it, and hers was really cool.
Yeah. Well, you can like opt out of things that you don't want to know because
Right.
you want to know if you're prone to like these types of cancers and these types of diseases.
Oh,
Stuff
that's
like
interesting.
that. So,
Yeah, but it's cool, and they can kind of do your lineage too. You know. So.
and tell you if you're related to celebrities.
Oh, that too. Oh, geez,
They
fun.
like swab their mouths at events, and then they can like opt in to where you can find out if you're like cousins or whatever with
Fun. It's crazy. It's crazy. The level of knowledge now. So the particular garden pea plant, the species that he used to study, Pistum stentonum. Gotta love these names. The good news is you guys don't need to know it, but just so that you know which one, right? Like I said, he was meticulous. And most scientists are, because they don't want anybody to refute their work, right? And so the reason why this one was really advantageous is that there were several varieties. So he could look at purple versus white colored, you know. You really can't study anything if every single plant looks pretty much the same, right? But for these, you had some variation. And they were distinct characteristics that you could easily visualize. Its structure allowed for easy crosses where the choice of the parent plants could be controlled. And this goes down to, you know, botany. You know, where are the sperm cells housed and where is the egg housed within that flower. They were discrete, separate units, so he could separate them. And so we'll take a look at that. So here is the portion where the ova are, where the ovary, where the eggs are. And then on the anther at the end is where the pollen granules, basically the sperm, are produced. So he could remove the male parts of the flower so that it couldn't self-pollinate. And he could actually physically do the crosses between different plants. Does that make sense to you guys? Great. Okay. So, pollen granules of sperm. Here are the eggs. So what happens is that the pollen granules land at the top on the stigmata, right? And then the pollen tube grows, and the DNA travels and eventually makes its way all the way down to the egg. And then you have the joining of the two, and you have the first cell of the new plant, which is the zygote. The endosperm, it sounds like sperm, right? But it actually is just food for the growing seed, or the food for when it does germinate. So he carried out two types of crosses. He allowed self-fertilization to happen, where the pollen from the same flower, right, fertilized itself, which it can naturally occur. Because of, like I said, the architecture, the way that plant is arranged, it can self-fertilize. He did cross-fertilization. I thought it was unlocked. Come in. Come in. It should be unlocked. By the spiral staircase. Yeah. Somebody's. What? This is like the third week, right? Yeah. He's probably going to beg to have his form signed, too. Get back into class. So this is where the egg and the sperm are from different plants, right, when we do cross-fertilization. So this is where, like, you know, the bee picks up the pollen from one plant, lands on the other plant. In this case, he physically did this. So he physically removed the male parts so they couldn't self-fertilize. And then he took pollen from the male parts from one plant and introduced it onto the female parts of the other plant. So he removed, right, the anthers, which contain the pollen granules. And then he took pollen, just simply like with a paintbrush, and took pollen and then put it on the female part of the other plant. That plant produced the seeds, right, the pea seeds, and then he also planted those to produce his first generation. And notice, he crossed white with purple, but he ended up with all purple. Now, he had started, too, with relatively pure, what we would refer to nowadays as pure breeding strains, where every time the white were bred with each other, he always got white. And all the purples, he had purples for generations upon generations. And then he started crossing those pure generations. So that's what we talk about when we talk about pure breed dogs, right? You can go back generation after generation, and they're all the same breed. The original breeds, by the way, were different crosses. And again, they just kept breeding the same groups to try and get those particular characteristics they wanted. So he started with what we call actually true breeds. So the morphological, what we're looking at, we call characteristics, right? So the purple flowers versus white flowers. The term trait describes the specific properties of that character. So eye color is a character, blue is the trait. So in the case of the plants, right, the character we're looking at is flower color, the trait is purple or white. So a variety that produces the same trait over several generations is termed a true breeder, or what we a lot of times call pure breeds. So the 7 characteristics. Well, one of them is definitely the color of the flower. Anyone know anything else? Well, they're pea plants. What do they produce as seeds? Peas. Do all peas look the same? No. Some are green and some are yellow. Some are round, some are wrinkled. The pod that holds them is either green or yellow. The pod itself can be smooth or constricted. The flowers, of course, can be purple or white. The height of the plant can vary dramatically, tall versus shorter, called dwarfed. Where the flowers are arranged, some of them are axial in position, others are terminal, meaning at the end. So those are the 7 characteristics he looked at. And as I said, again, he got lucky. Because as we said, some of the traits that we see, right, some of the characteristics are multiple genetic codes working together. But he got lucky. These were pretty simplistic, not multifaceted, and they weren't linked in any way to one another. You had a question? Oh, yeah. And there's so many different varieties of roses, too. Yeah. Different colors. And again, but it's all breeding, right? And then, staying within that group to keep producing that same result. And then crossing to get different results. And, it's over generations upon generations like we've done with dogs. They've done the same thing with plants. So what are the experimental advantages of using pea plants include which of the following? So who's been paying attention?
Well, they're all
Remember, read all answer choices. So yeah, all of the above, right? they were easy to cross, they were capable of self-fertilization, right? And there were several different varieties that he had to choose from.
So is the self-fertilization like the control group?
Yeah, you could say so. The term cross refers to an experiment in which so A, the gametes come from different individuals. That sounds like a cross, right? The gametes come from a single flower of the same individual. A is a cross. The gametes come from different flowers of the same individual. Would be the same, right? It's A. Is actually a cross. So as I said, at this point, I think everybody's using the ebook, huh? Or if you paid for it too, remember you could order a copy. Did anybody? I know one person did. Shay did, because she had told me how much she paid for it. Anyone else order to have one come in the mail? Okay. Have you gotten it? You just ordered it. Okay. All right. So otherwise, you can either click right here or click the library, right, and get to your ebook. Because everybody is going to have the ebook no matter what, because you got to pay for your Connect assignments. So navigation, right? We can go to chapter 2. And the different sections. So we're on section 1. It's already on 1. So guess where I got these questions? Literally right from your book. And with the ebook, you can click and it will tell you the answer. I did this to remind you that your book is an excellent resource. And the ebook is even better. You don't have to flip to the back of the book. You can just click. How cool is that? Alright, so let's see if we can get the next one right. I don't need one anymore to close that. Alright, so, because I didn't put my answers on here. To avoid self-fertilization in his plants, Mendel had to, he had to remove the anthers from the immature plants before they formed the sperm, right, and could, like we could have done with that stray dog, we could have removed his testicles so he wouldn't get my mother-in-law's dogs pregnant. Right? We thought they weren't active yet. Apparently they were. So just to be on the safe side, right? The correct answer is B. So the next thing that we need to be able to do is we need to be able to analyze his experiments involving a single factor cross, where we're looking at just one trait. One characteristic, one set of traits. Define the law of segregation and how it relates to gamete formation and fertilization. And predict the outcome of a single factor cross using a Punnett square. Yes, Punnett squares. Alright, so the law of inheritance can be used to predict the outcomes of genetic crosses. Right, so any animals and plants, breeders, are concerned with the types of offspring produced by their crosses, of course, right? So knowing how these things are inherited would help us in making the crosses we wanted and getting the desired offspring. So we want to be able to predict. And of course, this is important as it relates to genetic disease, right? So some people go and specifically they want to find out So there's, you know, some that are linked to ethnicity, right, because of all the inbreeding that's happened in the Jewish community. They have a genetic defect referring to as Tay-Sachs, right? And so if you're Jewish, a lot of times nowadays they'll go and you have genetic testing done to find out if either one of them have the trait and what is the probability that their child will have that disease. Yeah. They used to require back in the day people to go for blood testing, and I'm not sure what level of, like, what they were testing for with blood testing. I don't know if it was just blood types or how advanced it was. That has been discontinued. I don't know any state in the United States that does blood testing before marriage anymore. And quite honestly, you can marry your cousin if you want to. It's not advisable, right, to mate. No, I said marry. It's not advisable to mate with your cousin, right, because you have similarities, right? So again, that's inbreeding. You're going to amplify potentially bad traits, especially if you have particular diseases that run in your family. So it is better to be different. So I used to joke with my friends. They'd be like, well, why'd you marry a Southerner? I said, well, I was trying to help outbreed the South. So, I wanted to say something. I don't know, I heard on the news some crazy girl married her father, her genetic father. Like, they didn't grow up together. But then she married her father, but they adopted children. They didn't actually have children of their own. But yeah, that's still... It is. She did it. It's crazy. I don't know if anything as far as marriage is illegal anymore. Right?
You can't have more than one.
You can't have more than one marriage in most states, right? You can only be married to one other person at the same time. You can't practice polygamy.
You probably can't marry like your sibling.
I don't know. I don't know. I don't think so. I think it depends on what state. Look it up. Yeah. But, yeah. Yeah. But there are reasons why certain laws like that were on the books at one time. It's not a good idea. Genetically, it's not a good idea. Right? Okay. Moving on. Mendel did not have a hypothesis when he started. Like, he didn't have a best guess on what he thought was going to happen. Instead, he was very methodical, right, in what he did and recorded every step of what he did. And then, therefore, he was able to analyze it. He was able to quantify it and mathematically come up with an explanation for what he was seeing. Does that make sense to you guys? Right? So, I mean, he did going, clearly there's got to be differences, but he's like, there's got to be something, right? He's like, something is determining this. So this is referred to as the empirical approach. And so, we can deduce empirical laws based on this particular approach. So, of course, he studied, as I said, the 7 different characteristics, and they all showed just 2 variants. So, in comparison, blood typing, when we say ABO blood group, how many variants or alleles are there in the ABO blood group? There's A, B, AB, and O. There's 4 different possibilities within the population. Can you imagine trying to keep track of that? It's not easy, right? So again, he's lucky. He kept it simple. It was one or the other. So more like RH blood type, right? You either have it or you don't. That's much easier to study. So tall versus short or dwarfed. And at first he just looked at monohybrid crosses. So he just crossed two variants. He just looked at two things. Tall versus short. Tall versus dwarf. I don't like saying dwarf. Short. Smaller. I don't know. Right, so we just looked at a single characteristic at a time. So in this case, tall versus dwarfed. So he found that when he crossed tall with dwarfs, the first generation of offspring were all tall. And then when he crossed two of those hybrids, those two crosses, right, he just allowed them to self-fertilize, that he got mostly tall, but he did get some dwarfed plants. And of course, mathematically, he calculated this, right? He did a lot of crosses, y'all, right? He found that for that first generation, tall versus dwarfed, all of them for the first generation were all tall, right? And then for the second generation where he allowed self-fertilization, right? So the first ones mated with themselves, right? He got the variation back at a particular ratio, right? Mostly tall, right? About a 3-to-1 ratio. And then all the rest of them. He found that there was a trait that seemed to be more dominant, right? More prominent. So for these, it was tall, it was round versus wrinkled, yellow versus green, purple versus white, axial arrangement of the flowers versus terminal, smooth versus constricted, and green versus yellow pods. So F1 showed only one of the two parental traits, right? One of them was dominant, right? Kind of masking the other, what's going on here. And again, for all of those, right, it was a 3-to-1 ratio. When they were allowed to self-fertilize, and you got that second generation. The predominant one was predominant. It was 3 out of 4. But the less dominant trait, what we refer to nowadays as the recessive, came back. How did that happen? Right? This refuted the blending mechanism because you shouldn't have gotten the dwarf back, right? All of them should have been medium-sized plants to a certain extent. So there was some type of particle, though, right? There was something that was moving from one plant to the other and resulting in these results. So he postulated a plant contains 2 discrete heritable factors for a given character, one from each parent. Right? We know this nowadays, right? That you get one set for mom and one set for dad. But really, figuring this out in his time was pretty freaking amazing, right? That he mathematically figured this out. So the 2 factors may be identical or different. So clearly there was differences, right? Because we saw differences. But there are some things that were the same. So we had at least, 2 varieties, especially for what he looked at. So when the 2 factors of a single character are different and present in the same plant, one predominated over the other, right? And that's what we see as what we call dominant. And for some cases, that really is it. It's kind of like dominating the other one, right? Most of the time, especially like when you look at the color for the flower, right? Purple versus white. White is actually non-producing of the pigment. So sometimes it really isn't that it's dominating the other, it's that it's the only one producing, right? So you've got one gene that's producing purple and the other one's doing nothing. But that one gene is enough that it looks purple. But then when you end up with two genes that aren't producing anything, you end up with what? A white flower. No pigment. No color. Make sense? So that term dominant, sometimes that's true. It's really dominating. But sometimes it's just the only one that's producing.
If they both produce, would it be pink?
What do you mean?
Like if white produce white and pink make purple produce purple.
Oh, okay, no. Yeah, so I don't know if we're going to do this one this semester, we'll see. So for instance, pink versus red. Pink is actually just a lighter shade of red. So for some plants, the reason why they're red is they produce a lot of red color. Others are actually pink because they don't produce a lot of red color. So they're a lighter shade of red, they're pink. It's not that white and red are mixing, like you do when you do paints, right? To make pink with paint, you mix white with red. Does that make sense? In this case, it's using less red, and therefore you get pink. So imagine like when you're coloring, right? If you did a really light coloring of red, it may appear pink. Does that make sense? Or if you really lather the red on, it's clearly red. You experience this when you paint your nails, right? The color seems to change. It's just the amount of color affects how it looks. That makes sense? Okay. So that, too, like I said, some people kind of misconstrue what we mean by dominant too, and what that means and can mean. And then recessive, right, is pretty much what you don't see, right? It usually is a non-functional gene. It's not producing anything. So during the formation, the pairs of factors for a given character segregate randomly, so that half of the gametes receive one factor and half receive the other. So you have two copies, right, of all your chromosomes. One you got from mom and one you got from dad. When you go to give them to your babies, you don't go, okay, all the chromosomes I got from my dad, I'm going to give to all my babies. No, it's a mixture. It's random, right? You may give chromosomes 1 through 6 that you got from mom to your babies, and the other ones you got from dad to your babies. Like, clearly when my parents made me, most of my moms kind of predominated. I look like her.
Don't you get like mitochondrial DNA from your mom?
Yes. Your mitochondria are different than your chromosomes.
Okay.
Your mitochondria, that organelle, yes, you only get from your mom. And it does have its own DNA, its own ribosomes. And so any defects, you can blame those on mom, because dad did not contribute to that.
You said mitochondrial DNA?
You can trace your maternal lineage, because you get them from your mom. And that's how they trace maternal lineage, is through mitochondrial DNA.
They do that to me.
So this was his first law, the law of segregation. Now this one is pretty much always true. The next one, the independent assortment, not always the case. Because it really depends on what chromosome it's on, right? And he got lucky, like I said, like all of these are on separate chromosomes. So genes we've already talked about, right? Alleles are differences in those genes, right? So purple versus white, which is making pigment versus not making pigment. The different alleles in the ABO blood type are A and B and O. And they combine into the different ones. So you can be homozygous, where both of your alleles are the same, right? Both of what you got from your parents are the same. Homo meaning same. Or you could be heterozygous. This means that yours are different, right? They don't match. Now for O blood type, which is recessive, which pretty much you only have that if both of yours are non-functioning genes or alleles, then I know I'm homozygous. If you have A blood type or B, you could have just one A or just one B. Or you could be A, homozygous, or you could be heterozygous. You could have just one A and an O, but the A, again, is dominant over the O, which is recessive. So with the AB blood type, we don't know, unless we do crosses and figure out if you're heterozygous or homozygous. For AB blood type, what would you say you are? You're an A and a B. Are those the same? No, so you're heterozygous. Yours are different. The genotype refers specifically to your genetic code. Right? So that what would be on your gene. And so some of them we have, you know, we're not talking about the actual code, but we are talking about what it codes for. So in my case, I don't produce anything, right? But if you have the A blood type, you're making that protein we call the A protein. If you're B, you're making B. Your genotype and your phenotype for blood typing are pretty much the same thing. Or I shouldn't say the same thing. They seem very similar, right? So like I said, I'm definitely OO. That's my genotype, which makes my phenotype O. Do you see how it seems like the same? So it gets confusing. Okay. So, but someone who is AB, again, that's their genotype and their phenotype. It's because their gene determines what's on the surface of the red blood cell. But truly, when we're talking about phenotype, we're usually talking about that physical expression, how you appear. So, for the tall plants that were pure breeding, they put capital letters to denote it's a dominant trait. So he figured out, okay, tall is dominant, so we write it as a capital T. And this one was pure breeding, which means that it probably was homozygous. It had two T's, two capital T's. The dwarf plant, on the other hand, and of course we stick with T, we pick the letter for the dominant trait to be the letter. We write small k, lowercase, little t's to denote the recessive trait. And again, this guy is probably homozygous because he's pure, true breeding, always gave rise to dwarfed plants. But when we cross these, since we each have two distinct units, again, you're going to give one of those to your children. Well, all the children ended up being tall because all genotypes were the same, right? Because little dwarf plants only had little t's to give, and big, tall plants only had big t's to give. So all the plants are heterozygous, big T, little t. So this kind of makes sense too when you talk about blood typing, where you can kind of figure this out. So I usually use me as an example. I meant to draw a Punnett square, but I'll just quickly make a table. I'm trying to think. I need 4 across and 4 down, right? No, I need 5 because I need a label thing. Okay. No. I was right the first time. Anyways, I'll just use part of it. So I'm O. So I'm definitely have O's, right? So, I don't like this color. I hate that dark color because it's hard to see. I need a Punnett square, which is just that. So delete. Oh, Erica. Just do it again. I need 3 by 3. I'm out of time, aren't I? Okay. But read, right? I'm going to post some ones for us to do next time. I think I might even do us some handouts. If you know your blood type, and you have children and you know their blood type, or if you know your blood type but you don't know your parents' blood type, call your parents, ask them if they know what their blood type is, so you can do a real world cross, figure out about blood types.